So let's look at immunity. Well, first, kind of like, let's just define what immunity is. And it has a pretty simple definition. Immunity is basically just your ability to keep from getting sick. It's the capacity to resist and defend yourself against pathogens. And so let's look and talk about what a pathogen is. So if you look at a pathogen, gen means to create, and pathos means disease. So basically a pathogen is anything that can create a disease in a person. And so it could be something like a bacteria, it could be a virus, it could be even a fungus, it could even be a poison or a toxin. Those things also create situations similar to disease. So if we look at a pathogen, this could be a pathogen here, and this actually is the flu virus. So if you look at this flu virus, when you look at it, even though viruses are very, very, very small, when you look at them, look, they're not exactly the same everywhere. If I look here, that looks different from that, which looks different from that. Well, the difference between a pathogen and an antigen is that the antigen is any part of the pathogen, any molecule, any particle that can be recognized as non-self. And as it's recognized as non-self, then our immune systems will react to it. So if this is something that you never find in the human body, we can react to that. If this is something you never find in the human body, you can react to that. So anything you can react to is an antigen. And the word antigen, gen, means to create. And antigen, or sorry, anti refers to antibodies. So in other words, anything that will create antibodies in the body are antigens. And so what's the difference between a pathogen and an antigen? Well, look, this whole thing, the red thing, is a pathogen. But look, there are different shapes sticking out off of this pathogen. And so here's a shape over here, and here's a shape. If these shapes are foreign to the body, in other words, they don't exist in the body, and they're non-self, they're not part of you, then we can create an antibody against it. So here's an antibody that was created against that. We're going to call it antibody A. But look, there's a different shape over here, and we can create a different antibody to it. And it's antibody B. Same thing over here, different shape. And that would be antibody C. So look, these are all shapes. And shape is actually the most important thing when we talk about an antigen, because it's a shape that has to be unlike anything in your own body, unlike you, unlike self. And so when we start talking about it, we can talk about self and non-self. And so if it is self, that means it's part of you, it's your molecules, it's your proteins, it's your everything. If it's not part of you, if it's not your protein, it has a different shape, then it's going to be non-self. And hopefully the immune system will react to this and it won't react to this. Any questions about that? So if we look at this, again, here's that flu virus, and look at all these different shapes that are on it. So if you were to somehow be injected with this, or somehow get it through the, I don't know, a sneeze or something like that, look, you could make an antibody against that. So that same antibody would attack that and that and that and that. These are the same shapes. But this is a different shape. This is a different shape. And so I would make a different antibody against that. So here would be antibody A, and here would be antibody B. And then look, there's another shape over there. And again, if it's foreign to you and it's non-self, it can be an antigen and you can create an antibody to it. Does everybody understand the difference between pathogen and antigen? Any questions? So, what do you think would happen if I were to like chop this pathogen up and I inject you with this, or I inject you with that, or I inject you with that? Well, look, I've chopped it up. In other words, I've killed it. So the pathogen's not able to infect you anymore. But look, this piece still has that shape on it. And if I inject it into your body, you're still going to make this antibody C against it. But are you going to get sick if I inject this into you? Will you get sick? Remember, this is not the whole pathogen. It's just a piece of it. And this piece is not infective. And when we get to talking about vaccines, you're going to see that's exactly what a vaccine is. It's either been killed, inactivated in some way, or it's just pieces that have been injected into a person. So let's look at how all this works. So remember, immunity is your ability to resist disease, but actually we have two types of immunity. One type of immunity is called innate. Innate means you're born with it. So the day you're born, you have this. And innate is also called nonspecific. And the reason it's called nonspecific is that it doesn't do anything any different for any different pathogen. It works the same way every time. And in reality, this is sort of a primitive kind of immunity. But we all have it. But we also have this other kind of immunity, which is called adaptive. Adaptive means it can change. It can change. And what it can change to is to fit the pathogen. In other words, it can become different to fit each pathogen. So it's also called specific, because what we're going to make is specific for a particular pathogen. And so that word means it's going to work differently for every single pathogen. It's going to work differently. Not like this one. This is always going to work the same, no matter what the pathogen is. And so, like I said, this is kind of primitive. Well, this is much, much more advanced, because it works differently, specifically for each individual pathogen. And so we're going to look at both of these, but we're going to look at innate first. So let's look at innate. Again, it's also called non-specific. So innate or non-specific has some advantages to it. One of its advantages is that it's always there. It's very, very fast. It's always present. And because it's always present and ready to go, it's very, very, very fast. Another advantage is that you don't need to have been exposed to this pathogen in order for it to work. It's going to work against all pathogens. It's going to work against all of them, a wide range of pathogens. And so that's a huge advantage also. But it also has some disadvantages. And the biggest disadvantage is that it has no memory. No memory. Well, if you don't have any memory, that means you cannot learn anything. If you don't remember anything from A and P, you didn't learn anything from it. And you can't take any skills with you to anything else. If you have no memory, you can't learn, which means you can't get better. And so it's going to work exactly the same way every time. It does not get any more efficient if you're exposed to a pathogen over and over and over, because it works exactly the same way every time. So that's a huge disadvantage. No memory, does not get any better, works the same way. So let's look at some of these innate defenses. So if we look at them, here are the innate defenses, and you can divide them up into two groups. Those that are on the surface, in other words, they're facing the outside. And then those are on the inside, if something gets across this surface and inside your body. So let's look at surface barriers first. Surface barriers are basically walls. So look at this. This is the Great Wall of China, and it was built to keep the Mongols out. But think about this. Here comes a Mongol, tries to get across it. It is always there. They built it. It's there. And so it's always going to repel the thing. But look, it's not going to treat a human any different than, say, it would treat, I don't know, a horse. So if a horse came up here, that barrier is going to work exactly the same way against the horse as it does against the person. And every single time this person comes up, this barrier is going to work the exact same way. It's not going to change. It's not going to get any better at repelling this person. And that's exactly what happens with surface barriers that we have. Skin and mucous membranes. Skin and mucous membranes are always there, which means they're very fast. They're effective. They're our first line of defense. Remember, skin has keratin in it, which makes it tough and dry. Mucous membranes, remember, they have mucus, and some of them also have cilia. And so they can push things away and move it along and make it and it can get stuck in it. So skin and mucous membranes are these surface barriers. And as long as nothing gets across this, you don't go any further. So if you look on a person's skin, there's tons of bacteria that live right there on top of the skin. But they don't make us sick because they can't get through. They live on the surface. Now, if you cut yourself, those surface bacteria can then get on the inside. And so now we have to have something else to defend ourselves, and that's where these internal defenses come in. So if we look at the internal defenses, there are cells which roam around looking for things that are not supposed to be there. And they are phagocytes. They eat stuff. Phago means to eat. The most important of the phagocytes are called macrophages. Phage, again, means to eat. And macro means big. They're big eaters. They eat a lot of stuff. Some macrophages wander around. They go throughout the whole region looking for things that are not supposed to be there. But some of them stay in a particular organ. And we've talked about some of these fixed macrophages before. But here's what a macrophage does. Look, it eats stuff. So there's a microbe. What this phagocyte's going to come up to is going to stick to it. These are like little arms that it sends out. They're called pseudopods. In other words, fake feet is what they are. Fake feet. But that's what they kind of look like, arms or hands or feet or something. And then it surrounds it completely. And what it's going to do now is it's going to pull that to the inside. And so it's in a little package, a vesicle. And then what's going to happen is we're going to take a lysosome. And if you remember lysosomes from general biology, they have enzymes in them, hydrolytic enzymes. And remember, enzymes, hydrolytic, means they break things apart. So we're going to combine these two together. And what's going to happen is the enzymes are going to chop it up. Well, when you chop something up like that, you kill it. So you've killed whatever it is. And all of this now is just garbage, just what's called residual stuff or residual bodies. And most of that is just going to get ejected. So all of that indigestible material, at least most of it, gets ejected. Any questions about that? We also have these other cells, which are called natural killer cells. Most of the time they're abbreviated NK cells. Natural killer cells can determine if cells are altered in some way. In other words, there's something wrong with the cell. Could be that it's infected with a virus. Could be that it's become a cancer cell. Well, once it finds one of these altered cells, it kills it. It's a killer cell. And the way it kills it is by releasing some compounds called perforins. Well, look at this word. It looks like the same word as perforate. And that's exactly what it does. These chemicals perforate the membrane, the cell wall. So what's going to happen is we're going to create a lot of holes in the membrane. And the holes, fluid is going to move in and particles are going to move out, and this thing's going to basically explode. And so when you look at it, here is our normal cell. Look, there's nothing wrong with this cell. Nothing wrong. And so a killer cell, when it comes up to it, it sees that there's nothing wrong because there's nothing odd being displayed. And so it's not going to do anything to it. It's not going to do it. Nothing will be killed. But when it comes to this cell, look, this cell is different. This cell is a cancer cell and it displays something odd. That is not normal. It doesn't look like this over here. It's not normal. And when the killer cell sees this, it releases those perforins and kills the cell. This is actually happening right here. There's a natural killer cell right there. And here's a cell that's altered in some way. Well, look, they come up and they get really close. Look, they're getting close. They touch the other cell. In fact, this is nicknamed the kiss of death. Because what's going to happen is it's going to analyze the surface proteins on this cell. It's going to determine it's altered. And it's going to release those toxins and things like perforins. And when it backs away, this cell is going to explode. So, those are phagocytes and NK cells. We also have proteins that float around in our plasma. These proteins are made by the liver primarily. And there's more than these, but we're going to only talk about two, and only in very brief. But these are the two that we're going to talk about. These are the most important ones. Well, one of them is called interferon, and that word sounds like interfere. Well, when you interfere with something, you prevent it from happening. And that's exactly what they do. Interferons prevent viruses from reproducing. They prevent the virus from reproducing, and so slowly the virus amount goes down because it can't reproduce. Now, something else is going to be responsible for getting rid of the virus, but this interferon slows down or prevents reproduction. The other one is called complement, and it's a strange name. It was called that because before people understood it, that researchers knew that there was something helping white blood cells, helping the immunity. They didn't know what it was, they just knew it was complementing it. They were adding to it. Well, what it turns out to be is a bunch of proteins, about 23 different proteins. And these proteins, what they do is they can link together like a jigsaw puzzle. And when they link together, they create holes in the membrane of bacteria. Well, again, if you're going to create a bunch of holes, fluid's going to go in, particles are going to come out, and this thing's going to explode. So it's similar to what the natural killer cell does, but this is not the same thing as perforin. It's a completely different type of protein. So we had phagocytes, NK cells, these antimicrobial proteins. Another one we have is called inflammation. And if you look at the middle part of this word, look, it's got the word flame in it. Flame, like fire. And if you think about the word fire or a flame, most of the time it's red and it's hot. And that's exactly what happens. What happens is we're going to get a wide response that's going to happen wherever body tissues are injured. It doesn't matter what kind of injury it is. It could be due to an infection. It could be due to something like you just bent it too far. You sprained your ankle. But when you sprain your ankle, tissues are injured. Or if you scratch something just a little too hard. Or it could be something like a mosquito bite. Or it could be a cut. Or it could be all kinds of things. Anytime that tissues are injured, what happens is those tissue injuries, they give off chemicals. And those chemicals are going to cause a bunch of things to happen. What's going to happen is a bunch of things are going to come to that area. A lot of white blood cells are going to come there. Once they get there, those white blood cells are going to be walled off. We're going to build a wall around the injury with the white blood cells on the inside. And then we're going to try to kill or remove or destroy whatever's causing the problem. And then finally, we're going to help repair. So if you look at this, it looks like this. So look, someone stepped on a tack, cut themselves some way. Remember, all these bacteria out here now have a way in, and there they are. Now that they're on the inside, they can cause all kinds of problems. And so what's going to happen is these tissues, which were injured, are going to release all these chemicals. And those chemicals, again, are going to cause a bunch of things to happen. One of the things that happens is going to attract white blood cells. So white blood cells are going to find their way here because they're attracted to those chemicals. They actually squeeze out. They attach themselves to the capillary and they squeeze out through the little pores. When they attach themselves, it's called margination. And when they squeeze out, it's called diapedesis. That word actually means walking across. So in other words, they somehow walk across the membrane. They squeeze out through the little pores. And they're going to rush to that area. At the same time, this capillary is going to become more leaky. So if it becomes more leaky, fluid is going to come out. So fluid is going to come out too. And when fluid enters the tissues, we're going to get just them swelling up. Not only will fluid leak out, but so will proteins. And one of the proteins that's going to leak out is what we saw in blood. It's called fibrinogen. Do you remember that one? Fibrinogen. I didn't spell it right, but anyway, fibrinogen. And once fibrinogen gets out here, it gets converted to fibrin. And so we're going to form sort of a clot. But it's more like a wall. It's more like a walling off. And so a whole bunch of things are going to happen. There's our tissue injury. Here are all these chemicals, White blood cells are going to come, and white blood cells are going to eat and destroy and try to get rid of things until we clear the area. We're going to get vasodilation. More blood is going to come to the area, so we get more nutrients and oxygen because we want to be able to fight. And these things need energy. And then we're going to increase capillary permeability. The fluid's going to leak out. Not only will fluid leak out, but so will these proteins. We're going to wall it off, and we're going to form sort of this patch to set the stage for healing. But look, there are 4 things that are always going to happen with inflammation. There are 4 what are called cardinal signs of inflammation. And the 4 cardinal signs of inflammation are these. We're always, that means in every single case, we're going to get heat. That happens because more blood comes, and blood is hot. It's going to turn red. That's because the blood is red. And then we're going to get leaking capillaries, so we're going to get swelling. And when that leaky capillaries and the swelling is going to compress nerves, and that's going to cause pain. So we're always going to get heat, redness, swelling, and pain. And then if it's near a joint, you also get loss of mobility. But it happens whether there's bacteria, but it doesn't have to be bacteria. It could just be something like a sprain or something. So look, every single one of these is something different. I don't know what happened with these people, but something is different here with every single one of them. But what is the same is inflammation. And if you look, you can see those 4 cardinal signs, or at least you can imagine them for sure. But look how red these spots are. You can see the swelling here, and the swelling here, the swelling here. And then remember, you're going to get compression of nerves, and so this is going to hurt. It's going to hurt. And if it's near a joint like this is, this toe is probably not going to bend. We're going to get loss of mobility also. So that's inflammation. There's only one more thing that's part of the innate defenses, and that's fever. So let's look at fever. So what is a fever? Well, basically all it is an abnormally high body temperature. So if you think about a body temperature, if a normal body temperature is 98.6, body temperature goes up and down. If you exercise, your body temperature is going to go up. But that's a normal thing. It's normal for exercise to make your body temperature go up. So fever has to be something different. It's abnormally high. Now, it doesn't have to be very high. You can have a fever at 100, or a fever at 100.5, or 101, or whatever. All of these are higher than this, and it's an abnormal high. Well, what causes a fever are chemicals called pyrogens. Remember, gen means create, pyro means fire. So it creates fire in your body. Most of us, as soon as we get a fever, immediately you're going to run to your bathroom cabinet or someplace like that, and what you're going to do is you're going to take something like Tylenol or aspirin or ibuprofen, because we don't like fevers. But in reality, fevers are actually good. If you're sick, having a fever is actually a good thing, because it does a bunch of helpful things. It enhances your immunity. It speeds up the cells. Things like phagocytes, they work faster. Enzymes work better. It accelerates tissue repair, healing. And so instead of taking a fever reducer, you probably should just live with it and go rest and recover, because fevers make you feel bad. So you go somewhere and you lay down and you cover up and you get warm and you just sleep and you just rest. Now, fevers can be bad if they get too high. So if they get anywhere around this or above, they're going to be bad because they start denaturing enzymes and they start killing off neurons. And so you can have things like seizures and so on. Most of us are more worried about children with fevers than adults, but that's actually the opposite of what you should think about. Children are a lot more resistant to the damage that fevers cause. So 104 in children is not something that's very, very dangerous. For a child, it has to get even higher in order for things like seizures and stuff like that to happen. So those are the innate defenses. Any questions about those? So look, every single one of these is in this innate category. And remember, the advantages of the innate is they're fast. These things are always there. They're fast. And remember, you don't need any prior exposure in order for these to work. Your skin works whether it's ever seen a bacterium or not. These work. But remember, there are these big disadvantages. And the biggest disadvantage is there's no memory. No memory. So we have advantages and we have disadvantages. So the question is, inflammation. It sounds like this is good for you. It's the body's innate defense. Why are we told to take anti-inflammatory medication and so on? Well, there's a good reason for that. Now, if you are sick, inflammation is good. But what happens is that cut right there, we're going to rush to that area. And so, yes, that is a good thing because we're going to kill off the bacteria. We're going to wall it off so these white blood cells can do their thing. But here's the problem with inflammation. Inflammation, it's kind of like if you decide to remodel your kitchen. So think about it. If you remodel your kitchen, the first thing you have to do is remove the old stuff. So that's what happens with this inflammation. But think about it. If you're in your kitchen and you're swinging a sledgehammer around, do you think it's possible that you might destroy something that's not what you want to remove, that you might have an accident and break something you really wanted to keep? Well, that's exactly what happens with inflammation. Yes, it's a good thing. But what can also happen with inflammation is it can destroy healthy tissue. So if you have an injury, if you have an infection, inflammation is good. But chronic inflammation is something that lasts for a really long time, and chronic inflammation destroys healthy tissue. And you don't want to do that. I mean, back in AMP1, we saw some of the things that chronic inflammation can cause. Remember the things like arthritis, we talked about that, which is a destruction of the joint. We talked about other things as well. Aditi says she can't hear. Can everyone else hear? So what you might want to do, Aditi, is back out and then come back in again. Make sure your speakers are on also. Anybody else have a question? Comment? Anything? Okay, so that's all innate. Again, remember innate, we're born with it. There are advantages and there are disadvantages. Well, remember, innate is kind of primitive. The reason it's primitive is because all animals have these things. And it's primitive because it doesn't learn. It cannot learn. And so it's pretty much a primitive system. But we have this other system too. And remember, it's called adaptive because it can adapt. It can adapt. It can change. And what makes it change or what makes it adapt is what it learns. So in other words, it has memory. It learns. And so if you look at the adaptive immune system, it's this functional system. You can't really see it. It's not like an organ, but it has some properties. And here's some things it can do. And one of the biggest and most important things is it can tell the difference between self and non-self. It can tell the difference between self and non-self. Well, how? First of all, what is self? We talked about it a little bit. Well, what is it? Self is everything that's you. Every single thing that's you. That means every cell, every protein, every carbohydrate, everything that's you. So what is non-self then? Basically, non-self is everything else. So I don't have to experience the whole world to know what non-self is, because if it's not me, then it's not self. So everything else is non-self. So if I take cells out of one person and put them into another person, here that was self, but it's got different proteins in here. So over here, it's going to be non-self. If I take anything out of the environment in here and put it in here, it's not part of me. It's non-self. And so that's the most important thing that this is able to do, is able to distinguish self versus non-self. And so that allows it to recognize foreign substances. Because if you know what self is, you also know what non-self is. And then it can enhance or amplify some of the things of the non-specific, some of the things of the innate. And one of those things, remember, was complement. So it can increase the efficiency of complement. And then finally, it's all about B cells and T cells. And that's what we're going to spend a lot of time talking about. So, just like before, we had advantages and disadvantages. Same thing here. We have advantages and we're going to have disadvantages. So let's look at those. So let's look at the advantages first. So remember, it has memory. It can remember. And it can remember the pathogen. It can remember the antigen. Another thing it can do, though, it can adapt. Even in its name, it can adapt. And so it destroys specific pathogens in very specific ways. And so if I have two pathogens, it's not going to attack them the same way. It's going to be very specific. Again, that's in its name. And then probably one of the most important things about it is because it has memory, and remember the antigen, it produces immunity that's long lasting. So usually you don't get sick from the same pathogen twice. If you have a subsequent exposure, it usually does not cause infection. And then finally, something that's also very important is if the injury happened on your toe, so right there, I'm going to make products, antibodies, and things like that right there. But it's not going to stay right there. Those antibodies are going to circulate through your whole body, through your foot to the other foot, up the hands, up to the head. And so they're going to protect the entire organism. So again, advantages: protects the entire organism, very specific pathogens in specific ways, has memory. And because it has memory, we produce this immunity that usually lasts a long time so that subsequent exposures to that same pathogen don't cause infection. They don't make you sick. So it sounds really, really good. But there's some disadvantages. And the disadvantages here, one of them is that it's slow. It has a lag time. Look how long lag time is. It's a week to a week and a half, 7 to 10 days. And during that 7 to 10 days, we're not doing anything against the pathogen. So the pathogen is going to multiply like crazy. And during that 7 to 10 days, what's going to happen is you're going to get very, very sick. But toward the end of that 7 to 10 days, we're going to start making products, and the products are going to kill the pathogen. And then slowly then you're going to get less sick until you're finally well. Another disadvantage is it's specific, but it's so specific that if you change the pathogen in any way, just tiny little changes in the pathogen, and you lose the immunity. You lose it. And if you lose the immunity, you have to start all over again. So you have to go back. You're going to have this lag time again. You're going to get sick again, and so on. And then, not very often, but sometimes mistakes get made. Remember that the whole thing is able to tell self from non-self. And this is supposed to attack non-self. It's supposed to attack and kill it. It's not supposed to do that to self. But sometimes a screw-up can happen, and your immune system begins to attack your own body parts. And so you wind up with something called an autoimmune disease, where your own immune system is killing you. It's destroying your own tissues because it doesn't recognize them as self. Any questions about any of that? The advantages or disadvantages? Well, remember that it's all about B cells and T cells. And so when we look at the B cells and T cells, it's sort of like having two arms of immunity, or two sort of branches of immunity. And B cells and T cells are going to be in charge of different branches of the immunity. So Devaney says, could that happen like out of nowhere? Well, what happens is it forgets. So it's not supposed to happen, but just like anything else in nature, a mistake can happen. And so it could be just a single cell that forgot or didn't do what it was supposed to do in terms of self versus non-self. that one cell can actually multiply and create havoc. So this is where things like rheumatoid arthritis and things like that come from. Some of the things we've already talked about. We'll talk more about how this can happen in a little while. But anyway, let's go back to this. So we're talking about two branches of immunity. And the two branches are controlled by different types of cells. We have two different types or two branches of immunity. One is called humoral. Humoral means it floats around in your plasma. It's in your fluids. It's also, and this is a newer and probably a better name, antibody-mediated immunity. So B cells are in charge of antibodies, and antibodies float around. That's why it's called humoral. So it's all about B cells. The other branch, though, is called cellular immunity, or sometimes now cell-mediated immunity. But in this case, it's not antibodies. Antibodies are proteins, they're not cells. So this branch has cells that are actively involved, and this is controlled by T cells. So two branches, humoral, which is also called antibody-mediated, and cellular, which is also called cell-mediated. And we're going to look at each one of these, and we're going to spend a lot of time talking about each one. Any questions to this point? Okay, well, remember, one of the main things about this part of immunity is it can tell the difference between self and non-self. Well, how do we know what self is? We have markers that determine self. They stick off of our cells. Guys, don't click on the PowerPoint. So we have markers. Well, the markers are proteins is what they are. And they're controlled by your DNA. So you got DNA from your mother, you got DNA from your father, and the DNA then is going to control this protein, and it gets expressed on all your cells. Well, the protein is called MHC. And here's what it stands for: major histo, which means tissues, compatibility complex. Another way to think about MHC is to think of it as your tissue type. So you probably heard about people who are on a transplant list and they're waiting for a donor. Well, you can't just use any donor. It has to be a tissue match. In other words, it has to be as close to this same person as possible. Now, it's never going to be perfect unless you have an identical twin. That's the only way it's going to be exactly the same. So if you happen to be an identical twin, you're very lucky, because if something ever happens to you and you need some sort of transplant, you have a place where you can get it and there will be no chance of rejection. But most people are not identical twins. And so if I take this MHC out of one person and I put it in another person, it's not going to be exactly the same. There may be some little difference. And that little difference is enough to have our immune system attack it. And so if you do have a transplant, what you have to do is you have to suppress the immune system. So for the rest of this person's life, they have to take immunosuppressants to keep this chance of rejection, chance of destruction down. Because it's not a perfect match. Again, like I said, unless you have a twin. So when we look at MHC, there's actually two types of MHC. Everybody has two types. One type is called MHC class 1. So if you look at MHC class 1, it's found on all nucleated cells. All nucleated cells. So every cell in your body that has a nucleus is going to have this sticking off of it. And there's going to be a bunch of it sticking off. So all nucleated cells. So let me ask you a question. Do you think you have MHC class 1 on red blood cells? Do you have it on red blood cells? Yes or no? So Doris says no. Why not? Why don't you have it on red blood cells? Because red blood cells don't have a nucleus, which means that this is not present on red blood cells. And so even though we have some problems with red blood cells, remember the Biol and all that, we don't have this problem. And so we don't have tissue rejection of red blood cells because there's no MHC on them. So we don't have to worry about tissue rejection. But any other cell, any nucleated cell, is going to have MHC class 1 on it. But that's not the only kind we have. We also have these special cells, and we'll spend a lot more time talking about them in a little bit, but they're called antigen-presenting cells. Well, look at the name. Their name actually tells you exactly what they do. They present antigens to the immune system. They present antigens. And so antigen-presenting cells actually take antigens and take them and show them off. They present them. Basically, they present them to T cells. We'll see that a little bit later on. But antigen-presenting cells have a different type of MHC in addition to MHC class 1. Remember, they're a nucleated cell. All nucleated cells have MHC class 1. But antigen-presenting cells also have a different one, and it's called MHC class 2. And they're the only cells that have MHC class 2. They're the only ones. But again, it's controlled by your DNA, the shape of it is, and all that kind of stuff. So that's how we tell self, is by these markers. And if a cell doesn't have the marker, or if something has happened to the marker, then we know that there's something wrong with the cell, and we can destroy the cell or get rid of it. We'll see that again a little bit later on too. And then if we know self, we also already know non-self. And so non-self, again, is going to have anything that's different than that MHC. And we're going to build something against it. We're going to build something against it. And we're not going to build it against this part. We're going to build it against this. And remember, it has to do with the shape. And the shapes are what we're going to react to. So if we look at this adaptive immune system, there are three types of cells that we're going to be concerned with. We already talked about B cells and T cells, but there's also these antigen-presenting cells. So let's look at these. Antigen-presenting cells, what they do is sort of an auxiliary role. Their name, again, tells you exactly what they do. They present antigens, and they present the antigens to these cells down here, these T cells. And then these are the ones that are going to be most active in immunity. So they have a direct role, a full role. And a good way to think about this is to kind of think about the military. So just like the military prepares for war and fights a war and so on, that's what these cells do too, because it's just kind of a war going on against a pathogen, whatever it is. Well, think about the military. There are different branches of the military, and they work differently. And so we have different branches of our military too, and they work differently. If you look at the B cells are similar to how the Air Force works. Think about the Air Force. The Air Force, you've got planes flying up in the air. And so the guy that's doing the fighting is here, where his enemy's way down here somewhere. They're on the ground or someplace else, or on a ship or something like that, but there's a long distance between them. T cells are more like the Army. If you think about the Army, even if you have something like tanks or guns or battleships, you have to be fairly close to the enemy. And when we're going to see that exact same thing, T cells, they're going to come up to the cell. They're going to be very close, and it's a little bit like hand-to-hand combat. So we're going to see 3 different types of cells. So let's look at these antigen-presenting cells first. So antigen-presenting cells are a little bit like spies that gather information. And the way they do that is they eat stuff. They eat foreign particles. And then what they're going to do is they're going to take pieces of those foreign particles, little fragments, and they're going to carry them and show them off or present them from T cells. So they're sort of like interrogators or something. They gather information. They're all about information about the enemy, about the pathogen. And then we have B cells. Remember, they're a little bit like the Air Force. So think about the Air Force. So here's the Air Force. Here's a guy, and look what happens. They drop bombs. And those bombs are actually the part that does the killing. It's not this guy here. Even though he drops the bomb, he's not physically involved. He's flying a plane. And today, you don't even have to be in the plane. You can use drones. So he could be in, I don't know, Cincinnati or someplace and drop these bombs halfway across the world. Well, most bombs are kind of dumb. They kill all kinds of things. So if you drop a bomb on I don't know, an area of land, everything in that area is going to be destroyed. But that's not what it's like with B cells. They also make bombs, sort of, but they're smart bombs. They're called antibodies. And so antibodies are produced from a cell, just like the guy in the airplane. They're sent way out. Here they are over here, far from where it is. But they don't just kill everything. They're very, very, very smart. They're very specific. And usually they only attack one thing. And so they don't damage anything around it, just that one thing. And then T cells, remember, are more like the Army. So if you think about the Army is directly involved in the killing of the enemy, whatever it is. So look, with the Army, we're going to have lots of participants, lots of cells. And these cells are going to go marching out, and they're going to fight a war. And it's going to be close combat. Remember, the cells are actually going to go right up to the other cell and basically touch it before they kill it. It's hand-to-hand. It's close combat. It's not these little bombs. Remember with the bombs, this thing is not living. It's not a cell. It could be an antibody, but antibodies are not cells. They're proteins. But here, it's people. It's cells that are doing it. And these cells are going to physically destroy the enemy. And so they work in very different ways. Any questions? Wow, we have 28 people here today. If you're new, I hope you're able to hear and I hope you're understanding. If you have any questions, just type it in and I'll stop and answer questions if you have any. Okay, so that's what we've got. And so we're going to have antigens. Well, antigens are going to get into your body somewhere or another. They can come through the nose, the mouth, they can come through your skin, they can come through the digestive tract. They are lots and lots of ways in. And so at some point, they've crossed the barriers, they've escaped the macrophages, they've escaped the natural killer cells and all that kind of stuff. So this other part of the immune system is going to get involved. And so let's talk about the very first way that it gets involved. In other words, we're going to intercept the antigen. So the first thing that intercepts the antigen is antigen-presenting cells. So antigen-presenting cells are basically phagocytes, is what they are. Now we're going to see later on that B cells also are antigen-presenting cells. In other words, they have a dual role. But here's an antigen-presenting cell, and we already know what phagocytes do with antigens. There it is. Remember, they're going to eat it. They're going to send out these little pseudopods. It's going to surround it. It's going to pull it to the inside. We're going to take a lysosome and we're going to combine it. And the lysosome is going to chew up all the little parts of this thing. Antigen-presenting cells are going to do something different. Instead of, once this is all chewed up, instead of them spitting it out, what they're going to do is they're going to take little pieces of it. So this is that pieces of that antigen. They're going to combine it with MHC. So remember, MHC is that self-protein. And remember, it can be either MHC class 1 or MHC class 2. Antigen-presenting cells have both. Well, in this particular case, look, it's MHC class 2. And what they're going to do with that protein, that or the whatever it is of the antigen, they're going to combine the two. They're going to take that and attach it to the MHC. And then they take it and send it to their surface. So look, what this antigen-presenting cell has done is it took this antigen, took pieces of it, and now what it's going to do is it's going to show that part off. It's going to go and show it to a T cell. So that's the very first thing that happens. And it can happen in lots of different places. It can happen in the skin, happen in the intestines, but most often it winds up happening in one of these lymph nodes or in another of these tissues, lymphatic tissues. It could be the spleen, it could be, anyway, it could be tonsils, could be any one of them. But those antigen-presenting cells are there waiting. And they're waiting for this antigen to come along. And again, they're going to eat it. They're going to chop it up. They're going to take little pieces of it, attach it to their MHC, and display it on their surface. And then we're going to take that and we're going to show it to a T cell We're going to go running over to a T cell and say, look what I found. Any questions about antigen presenting cells? So that's what they do. Let's look at what the B cells and T cells do. Well, before we actually talk about what they physically do, let's talk about how they're different from other cells. Remember, these are lymphocytes. Remember, lymphocytes are white blood cells, round cell, round nucleus. Remember, they look like this. A little bit of cytoplasm. That's not a good picture, but a little bit of cytoplasm around the outside. They tend to be a round cell and a round nucleus, big nucleus. And just like all white blood cells, they're going to start out in the bone marrow. That's where they're going to start. And so when we look at what they do, here's where they are. They're in the bone marrow. Remember, red bone marrow is inside the spongy bone of cells. And so the stem cell, we're going to give it a colony stimulating factor, the right one, and it's going to turn into lymphocytes. And then a different colony stimulating factor is going to separate these into what are called pre-B cells and pre-T cells. Another way to say this is that they're naive. In other words, they don't know anything yet. They're not mature. And they cannot do anything. They're just not ready to do anything. And so what's going to happen is they need to mature. They need to sort of go to school. Well, it depends on if it's a B cell or a T cell where that happens. T cells leave this bone marrow and they go to the thymus. And that's where they mature. B cells, on the other hand, stay in the bone marrow. And in the bone marrow, that's where they mature. And then once they mature, they leave the thymus, they leave the bone marrow, and they enter the circulation. And they circulate through the blood, they circulate through the lymph, and they circulate through the lymph nodes. Any questions so far? Well, when they're mature, there's a word for that. And the word is immunocompetent. So they're not naive anymore. Now they're immunocompetent. And so immunocompetent cells are the ones that are out here. We'll talk about what happens when they become immunocompetent. Remember, it happens in different places. If you think about B cells are going to stay in the bone marrow, and that's where they're going to become immunocompetent. T cells are going to go to the thymus, and that's where they're going to become competent. And that's where these letters come from. B stands for bone marrow, that's where it happens. T stands for thymus, that's where it happens. Another way to think about it is to think that they're sort of like they're going to go to school. And so B cells go to a local college, a local university, but where T cells is, they head off across the country or somewhere, or they go international or something, but it's far away. And then they're both going to go to school. They're going to learn different things. B cells are, when they learn, remember, they're going to become in charge of this humoral immunity. And humoral immunity is all about antibodies. And then T cells, when they go to school, they're going to learn and they're going to become in charge of this cell-mediated immunity or cellular immunity. And remember, they're not going to make these bombs. They're going to sort of go into like the army, and they're going to do hand-to-hand sort of combat. So they're very, very different cells. Not only are they attacked differently, they also attack antigens in different places. B cells attack antigens that are floating around outside of cells. So here are cells over here, and here's the antigen. Well, if it's floating around out here, the B cells can make antibodies, and the antibodies can attack it and kill it. But antibodies cannot get inside cells. So if you have antigen in here, it's hiding. It cannot be destroyed by antibodies. And so what's going to happen is T cells are going to come along. They're going to recognize that this cell is infected, and they're going to kill the cell. And so when they kill the cell, they're going to kill antigens inside the cells. So outside the cells, inside the cells. And if you get rid of all of the don't click the PowerPoints. Whoever's doing that, please don't do that. If you, I lose my train of thought when it happens. So if we clear out all of the antigen that's outside the cells, and we clear out all the antigen that's inside the cells, then hopefully what we're going to be left with is no antigen. And once there's no antigen, then hopefully you're going to get well again. So they go to school in different places. They're going to learn how to do things differently. And again, they're going to become immunocompetent. Well, if we look at that process, part of that process of becoming immunocompetent is to develop what's called a surface receptor. And so the surface receptor has to do with shape. And so we're going to develop a receptor on the surface, and it's going to have a very particular shape right here. And the shape is what is going to allow it to interact with an antigen. Well, in our lifetimes, we're going to encounter many, many antigens. And they all have different shapes. So that means that we also all need different shapes here. Well, the way the different shapes come about, please don't click things. Whoever's doing that, it makes it really difficult. Okay, so remember, it's all about shape, and this shape is determined by our genes. So the shape is determined by our DNA. And remember, we get our DNA from our parents, from our mother and our father. So there's this long strand of DNA, one from each parent. Well, remember, this DNA is going to code for everything associated with us. Hair color, how tall you are, all this kind of stuff. But there's a section of DNA, which is called the VDJ region. It's right here. The VDJ region is going to determine the shape of the surface receptor. So what's going to happen is there's a bunch of these, and there's a bunch of these, and there's a bunch of these. But what's going to happen is we're going to choose one V and one D and one J. And that one D, one V, one D, one J is going to determine the shape of this receptor. Here's how it works. What happens is, again, we're one V, one D, one J. What happens is it's going to do this. The DNA is going to fold up like crazy, like this. And all these parts that are sticking out like this actually get chopped off. They're going to get removed and discarded. That DNA is going to be discarded. Usually when we think about DNA, we don't think about getting rid of it, but we do. We discard it. And what we're going to wind up with is one V, that one D, that one, and one J. And so that one D, one V, one J thing is what's going to code for the shape. Well, look, this is totally random. It's a little bit like the lottery. Think about the lottery. Lottery, in this case anyway, there are 54 balls in here, and they're going to bounce around, bounce around, bounce around. Eventually, 6 of them are going to wind up here. And so those 6 numbers, whatever they are going to determine the winning lottery this time. But next week, they're not going to change anything. They're just going to do it again. It's going to bounce around and again. But how often do you think the same 6 numbers are going to pop up? Do you think that's going to happen very often? No, it's not going to happen. Hardly ever. If it did, we'd already know what the winning numbers were going to be. Well, look, with 54 balls, that's all that's in here, 54 genes, if you will, you can wind up with 175 million different combinations of these 6 numbers. 175 million different combinations, which means that you only have a 1 in 175 million chance of guessing what it is. Because every single week, there's a different set of 6. Every single week, there's a different set, a different set, a different set, a different set, a different set. Well, that's what happens with these genes too. And this different set is going to give us the shape of the surface receptor. So look, a different set is going to give us a different shape. A different set is going to give us a different shape. And the likelihood of 2 of them happening together is just the same as having the same set of 6 numbers happen twice. It's very, very unlikely. But here's the thing. When you talk about genes, it's not 54 balls. It's not 54 genes. The average person has about 25,000 genes that code for these shapes of these surface receptors. So it's like trying to win the lottery if you put 25,000 balls in here and you waited for the same 6 to pop up. You wouldn't have a 1 in 175 million chance of winning. You'd only have a 1 in about 1 billion chance of winning. And that's exactly what it is. So there's about 1 billion different surface receptors. So if this is our B cell 1 versus B cell 2 versus B cell 3 versus B cell, 150 versus B cell 9,000, every single one of these, when it folds up differently, is going to have a different VDJ. A different VDJ, a different set of lottery numbers. And so we're going to wind up with a different shape of the surface receptor. And so if you look at this, look, here's B cell 1, B cell 2, B cell 3. I don't know what the VDJ is, but this one is different than this one is. And when you have this different VDJ, look, you're going to get a different shape surface receptor. And this surface receptor is what interacts with the antigen. So there's the antigen. Well, look at this antigen. Does it look like that would fit with B cell 1? Do those shapes match? They don't. What about B cell 3? Do they match? They don't. But B cell 2 just randomly has this shape. This guy won the lottery. And the lottery is that it matches this particular antigen. But next week, you're going to encounter a different antigen. It's not going to be this guy. You're going to encounter an antigen that's shaped like this. That's not a good one. That's shaped like this, or this, or this. Look, now this one doesn't match anymore. He didn't win the lottery. This one's not going to match, but this one, if I do better, would match. And so this guy would win the lottery for this particular antigen. Does it make sense to everyone? So here's what that surface receptor looks like. And look, you got a bunch of them on every B cell. Well, on a single B cell, they're all the same. But between B cells, they're always going to be completely different. The chances of having two B cells with the same shape receptor is very low. But on a single B cell, there's about 1,000 of these. And right here is where that antigen has to fit. So whatever shape matches that, this particular B cell then is going to be selected. It's going to win the lottery. Any questions? Well, remember, we have to worry about self versus non-self. So what if this thing right here was not an antigen? What if that was your own proteins, your own stuff, you? Look, it matches this B cell. And if it matches this B cell is going to go through steps until it gets ready to produce antibodies that are going to attack this. Would you want to attack this? Look, it's self. It's your own protein. Would you want to attack it? If you do, you're going to get an autoimmune disease. You're going to get sick. You're going to destroy some tissues. So we need to know ahead of time if these B cells will attack self. And so before they finish their school, before they graduate and they become immunocompetent, we're going to test them. And they're going to take 2 tests. These tests are positive and negative selection. Positive and negative selection. And here's what's going to happen. They're going to take these tests. So here's our B cell. It matures in the bone marrow, becomes ready, and then now we're going to take these tests. So here are the 2 tests. One is called positive selection. The other one's called negative selection. Well, the reason they're called that is because of what it is. Negative selection, negative means no or not. And negative selection says that these cells must not react to self. In other words, they must not attack and kill your own particles, your own self. Because if this happens, if they attack self, you're going to get diseases like autoimmune diseases, and you're going to destroy your own tissues. So you would not want that to happen. But there's a second test that they take, and it's called positive selection. Positive means yes, it means you should, it means you must react to non-self. You must react to non-self because if it doesn't react to non-self, it's kind of useless. It's not doing anybody any good. Well, if these cells fail either one of these, they're destroyed. They're killed. At least if you go somewhere to take an exam and you fail it, they don't pull out a gun and just shoot you. But that's what happens here. If these cells fail either one of these tests, they're killed. So that this won't happen if these are self-particles. This won't happen. And if these are antigens, this will happen. It won't ignore the antigen. Any questions? Now a few, are sort of inactivated. And a few, are re-educated. Their receptors, their DNA gets edited. So you get a rearrangement of their receptors. But 99.9% of the time, what happens is they're killed. You don't want those cells hanging around. If they do pass both tests, B cells leave the bone marrow and they go to some lymphoid tissue. They go to tonsils, they go to lymph nodes, they go to the spleen, and basically they just sit there and wait. And what they're waiting for is an antigen that matches. So remember we said when we looked at this spleen, the lymph node, every one of these little black dots is the nucleus of a white blood cell. Almost all of them are B cells. Now there are also macrophages in here and other things, but most of them are B cells. And they're just going to sit there and wait. Let's look at T cells and then we'll stop for today. T cells are very similar. Remember T cells? They're going to start in the bone marrow, but they're going to go to the thymus. And the thymus is where they're going to develop their surface receptor. It also is controlled by the DNA. Whatever you got from your parents. But the receptor looks different. The receptor is this two-stage or two-part receptor. And the two stages recognize two different things simultaneously. They recognize MHC, which is on your body cells, and they recognize an antigen. So if you look at them, they don't look like B cell receptors. Remember, B cell receptors look like this. They don't look like that. T cells, their receptor has two parts to it. And the two parts are side by side. They're connected to each other. And one part recognizes MHC, and the other part recognizes the antigen. So the MHC and the antigen are hooked together, and they hook up or connect to this receptor. So if you look at it, this is what the receptor looks like right here. And look, it's connecting to a cell with MHC. There's the MHC. And there's the antigen. They're going to hook up like this. So when you look at it, it looks like this. So look, there's MHC, and there's the antigen, and here's our T cell, and the receptor is going to connect to the MHC. That's where it's going to attach to the MHC, and that one's going to attach to the antigen. And so it looks like this. Two-part receptor. One part recognizes MHC, one part recognizes the antigen, and they're going to hook up. But before they can leave the thymus, before they become immunocompetent, they also have to go through those same 2 tests. And so remember, the 2 tests are positive selection and negative selection. And again, negative selection means you must not, negative, not react to self. And positive selection, positive means yes, you must react to non-self. And again, if they're not, they don't pass these tests, then they're killed. And then the other ones go out and they circulate. Any questions? So that's where we're going to stop for today. If you come back tomorrow, we'll pick up right here and we'll continue with immunity. I'm so glad that so many of you showed up today. I have a record of the people who showed up. So if you're here for the 10%, then it'll show. I'll have that record. But I'm going to stop recording now.